Right. Good morning, everyone. Welcome to another session of the Australasian Fluid Mechanics Seminar Series. It's my great pleasure to introduce today's speaker, Dr. Xiang Cheng, who is an associate professor at the Department of Chemical Engineering and Material Science at the University of Minnesota. Professor Cheng received his BS in physics from Peking University in China in 2002, and then obtained his PhD in physics as well from the University of Chicago. His research, he has won several awards, and his primary research focus includes bacterial locomotion, hydrodynamics of active fluids, rheology of colloidal suspensions, and dynamics of liquid drop impact processes. And today he's going to talk about locomotion of flagellated bacteria, from the swimming of single bacteria to the collective motion of bacterial turbulence. Over to you, Xiang.
Yeah, thank you for the introduction. Yeah, thank you also for the invitation to give this talk. My group study, we study fluid mechanics of particle system. This could be a big granular system, as I show you on the left, all the way down to a nanometer scale or colloidal scale suspensions. But in the talk today, I'm going to focus on our research on bacterial swimming, low Reynolds number flow. So before get started, I need to acknowledge people who really do the work. So the work mainly done by my three graduate students, Dipanjan Ghosh, Shashank Kama, and Zhengyang Liu. Zhengyang has already graduated. And also two postdoc, Yipeng and Shuo Guo. For senior collaborators, we have collaborated with Xinliang Xu at the Computational Science Center in Beijing, and also my colleague, Lauren Francis at the University of Minnesota. Our research mainly funded by National Science Foundation. But either them or me are the leading character of the story today. So today's the leading character is actually E. coli, which is probably the most studied organism on Earth. Everything is known about this small creatures, genetic, network and everything about this creature. For us, of course, we are interested in how this bacteria or this small creature swim at extremely low Reynolds number. Reynolds number we're talking about here is 10 to the negative 5. So generally speaking, for E. coli, they have multiple flagellas. So as I show you here with this TEM picture, you can see there's a 5 flagella on the surface, on the body of the bacteria. So when bacteria swim, what happens that, if you can see my cursor here, so each of the filaments and connect to a small motor embedded on the membrane of the cell. You can see this yellow part here. And each of the motor will rotate on its own. When all the motors rotate in the same direction, the filament will come together, form this rigid bundle. And the bundle rotate counterclockwise if you look from behind. And this counterclockwise rotation will propel the bacteria forward. The bacteria size, as I indicated here, the body is about 2-3 micron in length, about 1 micron in width. As I mentioned previously, that each of the filament connect to a motor, and this is a schematic of the motor. This is a really elegant machine. The size of this motor is about 50 nanometer. It's embedded in the membrane of the cell. And you have the stator part, which is outside, as well as the rotor part, which is inside. The motor really behaves like a stepper motor, can rotate at really high speed and rotate at 120 hertz. So I cannot do that with my finger, but rotate really fast. The motor is connected to the filament through a very flexible hook, so that hook can be bent in all different directions, that it can translate the rotation of the motor to the rotation of the filament. And I also mentioned that motor can rotate in both directions, counterclockwise or clockwise. When all the motor rotate in the same direction, counterclockwise direction, the bundle form. But once in a while, on order 1 second, one or maybe a couple of motors will rotate clockwise. As you can imagine, if you have this bundle here, if one filament rotates in another direction, the bundle cannot stay there. The bundle essentially will open up. When the bundle opens up, the body will randomly turn into another direction. We call this a tumble. You can see here in this middle picture. And then bacteria will reform the bundle again when all the motor rotate in the same direction after about 0.1 second, and they will go into the next run phase. So we call this type of motion as run and tumble. And the reason why they do that, as you can see actually from this microscopy video, Generally speaking, you cannot see the filament because the filament is only 20 nanometer. In this video here from Howard Burke's lab at Harvard, they put a fluorescent label on the flagella, so you can see the flagella from the fluorescent microscopy. But typically, you won't be able to see the flagella, you only see the body. Okay, so why bacteria do this wrong on tumble motion? Apparently, they have to do this for their own benefit. The reason why they do that is because chemotaxis. What happens is that when bacteria swim in the direction, let's say along a gradient where you see the increase of nutrients, you see a food, you can see the concentration of the food molecule get higher and higher. Essentially, they will elongate the wrong phase. So they will tumble less. They will turn their motor less frequently. And then they will just essentially run in the same direction and they tumble less. And overall, you get this bias, a Brownian motion, which allow the bacteria moving toward a food particle, give them the chemotaxis ability. So that's one feature that I'm going to use later in my talk. The second thing is very important for my talk here is, as you can imagine, when you have flagella foam bundle, as I show here with the schematic, it's very rarely that the axis of the bundle will line up with the axis of the body. I indicate here, but it's a misaligned angle I call alpha. Because of that, as you can imagine, if I have this bundle rotating, push fluid, and then I push the body actually will slide in a different direction compared to the axis of the bundle. And overall, what happens that even in the wrong phase, bacterial trajectory is not going to be a straight line. It's actually going to be a helical shape. I exaggerate the helical here. Actually, in reality, the radius of the helix is actually much smaller than the size of body. But to really show the helix, I exaggerate the size of the helix. But essentially what happens, the bacteria just turn in the helical trajectory going forward. And in 2D microscopy, when you do optical microscopy, there's a 3-dimensional helical shape become essentially wobble. You can see the bacterial body just wobble back and forth. You can see my finger on the camera. The body is just essentially back and forth moving like this. I demonstrate this from the video here. I know online Zoom talk, sometimes the video is not smooth. If you cannot see smoothly, just trying to listen my description, you can see the body essentially just going back and forth like that. We call that wobble motion. And if you look carefully, there's another bacteria coming from the right. You can see this bacteria actually gyrating when it's moving forward, really badly, doing this helical badly. So that happens when the angle here is close to 90 degrees. Essentially, the body does not going forward too much. It's really just gyrating while it's going forward. So this is called wobble. Again, another feature that I'm going to use later in my talk. So with that preparation, I'm going to tell you the outline of my talk. I'm going to tell you two things. One thing is really single bacteria swimming in complex fluid, higher viscosity fluid. The second thing is when you have a large number of bacteria coming together in a dense bacterial suspension, you can see this very fascinating turbulence-like behavior. So I'm going to talk these two behaviors. So let me focus on first one first, how bacteria swim in a complex fluid. The whole research actually is inspired by a work of my colleague, Ed Kastler, who retired just right before pandemic. In 2004, Ed Kastler, my colleague, asked this very interesting question. Will humans swim faster or slower in syrup? Okay, so compared to water and syrup has viscosity 2 to 3 times higher than water. When you have people swim in syrup, are you going to swim faster or slower? Right, very interesting question. So I'm not sure you know the answer. So first of all, I have to emphasize this is a very hard experiment. I think I read some articles, Ed has to obtain more than 25 permits to do the experiment. One of the permit has to ask a city of Minneapolis that when he finishes experiment, he can dump the whole swimming pool of syrup into the city drain without causing any environmental issues. So that's probably the hardest permit to obtain. And after he has this whole swimming pool of syrup, he asked the University of Minnesota professional swimmer, professional athlete to swim in water as well as in syrup, and then measure their swimming speed. So on Zoom, I think I cannot ask questions. Usually in person meeting, I'll ask a question and people will show hands what they think. But I will still give you a chance. You think about it on your own. Do you think people will swim faster or slower? Okay, give you a couple of seconds to think about that. Now I'm going to show you the result. By the way, this is a very hard experiment, but it's worth it. I'd get Ig Nobel Prize next year in 2005 because of this work, okay? The answer is that it doesn't really matter. So this is the result. At Reynolds number of 10 to the 6, if you normalize swimming speed of people in syrup by the swimming speed in water, no matter what style you use, butterfly, backstroke, breaststroke, freestyle, whatever the style you use, the swimming speed is constant. There's no dependence on the viscosity of the fluid, at least in this range. So basically the inertia you gain from the higher viscosity is balance the drag coefficient or drag force that you have in the system. So they balance out, you get the same swimming speed. What's really relevant to the talk here is that someone that read an article in Nature said that had Custer done his experiment on swimming bacteria instead of humans, he would have recorded much slower times in syrup than in water. Okay, the general idea is that when you have a bacteria, there's no inertia at all, Reynolds number 10 to -5, as I mentioned early on. Everything you have is essentially drag. If you have a constant torque output, which by the way is what happens for the Fajala motor performance. So this is the torque output of a Fajala motor I showed you early on as function of how fast you rotate your motor. As you can see, as long as your rotation speed is smaller than this value, about 2,000 Hz, in reality, you're always smaller than this value. The torque output of the motor is constant. Now, if you have a constant torque output, you're just increasing the background viscosity. Apparently, you will swim slower because if you draw the low line of your motor, this is a low line. The slope here depends on the viscosity of fluid. You have this cross point or the red dot here. This red dot will determine how fast you rotate. When you're increasing viscosity of background fluid, you're increasing the slope, then your red dot will go to a smaller value, then you rotate at a lower rotation speed. At low Reynolds number, everything is linear, Stokes equation. When you rotate slower, you move slower, right? Another analogy you can think about is when you're backing up hills, use the same force to back. When you go up hills, because the load is higher, you have to back slower, right? Your speed goes down. So that's all makes sense. But apparently when this guy writing this article, he didn't check the literature. Actually, back to 60s and 70s last century, people already studied how bacteria swim in higher viscosity fluid. In this case, they dissolve some polymer, in this case PVP, in water, which increasing viscosity of the fluid, and they plot velocity of bacteria swimming speed, as function of 1/over viscosity, fluidity. So actually go from right to left, you increasing viscosity. As you can see, for all different type of Fajala bacteria, especially for E. coli, this is a bacteria we use, you can see when you're adding a little bit of polymer into the system, when the viscosity increases a little bit, bacteria actually swim faster. The line go higher, non-monotonic. When you go put a lot of polymer into your fluid, the speed goes down. So this is really mind-boggling, right? Bacteria not only keeping the same speed like human being, they actually swim faster when you get higher viscosity in the fluid. Eventually, when you put a lot of polymer in, it will slow down. And there are a lot of work. After that, I only list subside of the work. I also show the result from two most recent work. People measure velocity of bacteria swimming as function of viscosity. You can see viscosity increase. Same here. When you put different polymer into the system, velocity increase when you increase in viscosity. And this is apparently very important. That's why so many people study that, because we know in reality, very rarely bacteria swim in water. In their natural habitat, no matter in our body, in ocean, in soil, there are always all different type of macromolecules, DNA, proteins, or colloid particle. So they always live in a non-Newtonian environment. The important thing is to understand how they swim in those type of non-Newtonian fluid with higher viscosity. That's why people do so many research on that. Apparently, this is a 60 years old or 70 years old problem. There must be theory to explain what happens. So at least the four major theory in the field. The first theory actually from Howard Burke himself, right after that experiment in the 70s. What Burke suggests is that when you add polymer into fluid, polymer forming this gel network structure with a mesh, bacteria actually swim through the mesh hole, through polymer. That allows them to swim in more straight line and give the higher velocity. And later on, someone put this into mathematical formula and show mathematically that indeed you can get some speeding. That's the most cited theory and on this topic. So more recently, there's more result on this. People argue that this gel network theory cannot be right, because when you put a polymer into fluid, when the polymer forming this gel network or entangled network structure, the size of the hole there should be on order of a radial gyration of the polymer molecules. For the molecule that they use in the experiment, the hole size should be on order of tens of nanometers. We know bacteria is 1 micron, as I showed you early on in the first slides. 1 micron particle cannot go through tens of nanometer holes. So this cannot be right from polymer physics point of view. Then the more recent theory really using the non-Newtonian constitutive equation of polymer solutions. One idea is that, remember the flagella rotate at 120 Hertz. So 1 second you rotate 120 times. The shear rate at this scale is really large. They argue that near the flagella, essentially you show shear thinning behavior. You shear thin the viscosity of polymer solution. The head, because it's much bigger, the head rotates at only 20 Hertz to balance the torque between the head and the flagella. So the head rotates at really slow speed. It's experiencing normal viscosity of the solution. So now imagine that you have a single bacteria, you have 2 different fluid around this single bacteria. Near the flagella, because shear thinning, you have much lower viscosity compared to the viscosity of fluid near the head. So they call this type of a picture as a 2 fluid model. And then you can do some calculation to show that when you have 2 fluid model, you can also have speed enhancement. Third one, along the same line, but instead of shear thinning, they argue that because the shear rate is so high near the flagella, you stretch out the polymer molecules near the flagella, because the polymer coil can be stretched out at high shear rate. Now, when you stretch out or deform the polymer coil, you get no more stress difference. It's like the classical rod clamping experiment. You put the rod in polymer solution, you rotate it, the fluid will clamp up, because no more stress difference. And this no more stress difference can also give you a speed enhancement. The last explanation is a pretty ironic explanation. This is suggesting that when they do experiment, they get a PVP from the same company and they dialyze the polymer, get rid of the small molecular weight contamination in the system, make the PVP much narrower distributed. And they found that the speed enhancement goes away, or at least drastically goes down. And because of that, they argue the reason why bacteria swim faster is because bacteria eat the small molecular weight contamination in the system. So they eat polymer and they get more energy and they swim faster. That's really not a physical argument, but biological argument why they swim faster. So when we get into the field, we don't want to get into this really a fight on what is the right theory. We think that a lot of people study polymer swimming in colloid. Let's study something different. Let's look how bacteria behave in a colloid suspension. We'll have a bacteria swimming in colloid suspension, and we want to see what happens. And it turns out that actually there's a lot of similarity between the two systems, and here, there's a lot of similarity between the two systems. So that's why I have this introduction there. But let me tell you what we found first from the experiment. The experiment itself is very simple. So it's a very easy experiment. You just have your colloid particle. The radius go from 100 nanometer all the way to 1 micron. And we vary the concentration of the colloid volume fraction from 0 to 20%. And then we put some bacteria in this case, just wild type E. coli, and very low concentration, so bacteria do not talk to each other. And we put them together into this microfluid channel, and then we can look how bacteria swim in the channel. Okay, very simple experiment. And here's the result. So the y-axis, I plot swimming speed of bacteria normalized by swimming speed in water. So by definition, when there's no particle at all, when volume fraction 0, the velocity is 1, right? So the first experiment I do here, I use a very small particle. In this case, 100 nanometer PS particle. So I draw the cartoon picture. This is real size ratio. And think about this. If this is a bacteria, the size particle is like that, okay? When you add a little bit of our particle in the system, you can see a very small enhancement in speed. At volume fraction about 2 to 3%, you see about 5% enhancement in speed. But then when you put a lot of particle into the system, the velocity start to go down, okay? Now, if I go to bigger particle, in this case, 200 nanometer PMMA particle, another type of a polymer, but hard sphere particle again, the size ratios of what I show you here. Now at volume fraction about 3%, you see this peak here, which give you about 50% speed enhancement. Quite a lot of speed enhancement. Now you add more particle into your system, increasing volume fraction, the speed goes down again. Finally, I go to this big particle that we have, in this case, 500 nanometer radius or 1 micron diameter polystyrene particle. Again, at very low concentration, about 4% volume fraction, you see this is a huge peak. The speed enhancement is about 80%. To give you the sense of 80%, how big it is, so I always use this analogy. So Shashank, who is my student run this experiment, this is his running speed. He's a very good runner. His running speed and running speed of world champion. That's how much we're talking about 80% in human skill. Huge enhancement, right? You can see that clearly from the video. Now, let me summarize what I'm telling you so far. Just remind you what happens. When increasing size of particle, I see stronger and stronger enhancement. You can see the peak value that higher and higher. But all the peak happens at low concentration of polymer, okay? So the highest volume fraction we go when we see peak is less than 4%. So at low volume fraction, you can see very strong enhancement. Now we can also plot same data in a different way. I'm plotting the peak velocity normalized by velocity in water, plot that as function of the size of my colloid. I normalize the size of colloid by size of my bacteria, which is always 1 micron. So this is make everything dimensionless, but essentially R is a constant, right? Small r is a constant. Now you can, as I showed you early on, we increase the size of particle, velocity get enhanced. You get faster and faster swimming in the fluid. But what's really interesting is when you compare this result with what you have in the literature for polymer result. So for polymer result, if you use a radial gyration of the polymer coil, you can see that all the data collapse together into a single line, okay? So it seems that there's some relation between the two, but this is not the only similarity that you see in the system. There's another interesting feature that you can compare. So as I mentioned early on, when bacteria swim, it wobble back and forth. It has go back and forth when it's going forward. And we found there's a strong anti-correlation between wobble and swimming speed. As you can see here, black line is the swimming speed. When the swimming speed goes higher, the wobble angle goes down. When the swimming speed goes down, wobble angle increase. So there's a very strong anti-correlation. And this also makes sense, right? When bacteria wobble too much, it doesn't go forward too much. When you swim in a more straight line, you're going forward faster. But what's interesting is to compare this with polymer. To compare with the polymer result, I directly plotting velocity as function wobble angle. Again, for my result, it's almost linear anti-correlation between the two. But then I get the literature result. There are only 2 studies on wobble angle as function of swimming speed. Again, they overlap with my result really well. So you can see this anti-correlation in polymer system too. Now, how about the shape or trajectory? Here's what I see in terms of shape of the trajectory. Without colloid, essentially bacteria in water, you see this is zigzag trajectory, short and zigzag trajectory. But as long as when you put small amount of colloid in the system, you can see the trajectory becomes smooth and straighter, right? And when you look at polymer system, this is a polymer system. Without polymer, this is the same as a bacteria swimming in water. You see this is a short zigzag trajectory. But as long as when you put a small amount of polymer into the system, you can see the trajectory becomes smooth and straight. And from that, you can back up a lot of different quantities. You can measure rotational diffusivity of your bacteria. You can measure the run time of a bacteria, tumble time of bacteria. You can measure lot of different things, right? And then you can compare the result between colloid and polymer in dilute limit. They're all very similar. So I don't go to detail here. There are a lot of data, but essentially we measure diffusivity, run time, and tumble time. They all show same trend as function of a concentration of particle or concentration of the polymer. So the conclusion here is that all the 7 reported swimming features are quantitatively similar in dilute colloid suspension and dilute polymer solution. So if you take this result, this is a purely experimental result, no mathematical proof here so far. If you think that all the report results are similar between the two. There must be a common underlying mechanism determining bacterial mobility enhancement in dilute colloid suspension and polymer solution. If you believe this conclusion, you can use this result to compare the existing model. Just remind you what we have, right? We use hard sphere colloid particle. When the volume fraction is less than 4%, you see more than 80% speed enhancement. The network's theory cannot work because hard sphere particle at full volume fraction, there's no way that a particle form any network. Actually, at this so low volume fraction, particles do not even touch each other. There's no way that they form any general network. So this theory cannot work. How about shear thinning? At volume fraction of 4%, even Einstein's relation for suspension viscosity still apply. You just apply Einstein formula for suspension viscosity, you see the suspension viscosity go about 10% higher than water. Now, let's say you shear thin so much, your flagella rotates so fast, you shear thinning so much, you get rid of all this 10% actual viscosity. Okay, so your body is in suspension 10% higher than water, your flagella is in water. You put it into any of these two fluid model, it's far from enough to explain the 80% speed enhancement that you can see in the experiment. So it's too weak to explain the strong enhancement. How about deformation of chains? Our particles 2 to 4 gigapascal. There's no way that the rotation of a small flagella can deform the particle. So there's no normal stress difference, at least from deformation of the particle. There's a very, very weak elasticity from the Brownian motion of the colloid particle. But again, that's just too small to be considered. So the third theory cannot work. The last one, apparently, we dilate our particle really carefully. There's no contamination in the system. And after that, I tried to search literature whether E. coli can eat polystyrene or PMMA polymer or plastics, essentially. And then my colleague in polymer, they tell me, you don't need to do the search. If E. coli can eat those polymers, we won't have this environmental issue on polymer in oceans, right? This micro polymers, plastic will be all degraded by microorganisms. So there's no way they can eat those things. So none of the theory really works if you believe there's a common origin of the speed enhancement. So we destroy all the theories. Now I need to tell you something that why they swim faster, right? Now let me tell you some of the theory we have. So imagine if you have a ball in water, you drag the ball from left to right with some force, and you get a velocity to the right. At low Reynolds number, we all know this is essentially Stokes law, right? F equal to 6 pi eta A V. A is the radius of the particle, Stokes law. But now imagine that you put this particle next to a wall, non-moving wall, because you have to specify the no-slip boundary condition between the particle and the wall. Even though you only drag the particle horizontally to the right, the particle has to rotate. It seems that there's a torque applied to the particle. We call that boundary-induced torque. It's not real torque. It's just because you have to satisfy no-slip boundary condition between the particle and the wall. The particle has to rotate, essentially have a small torque there. Now, same thing what happens when you have a bacteria swim next to a particle. Imagine you have a bacteria right now swim vertically. Because the distance between the bacteria and particle can be really small. Some experiments show that this distance can go down to 50 nanometer or 100 nanometer. Now you can have lubrication approximation to calculate how much torque the body will feel. Even though bacteria only swim vertically, because the presence of the solid particle, body will rotate to the right. And using lubrication calculation, you calculate how much torque you have there. The important parameter here is gamma, which is size ratio that I showed you early on, size of the particle divided by size of the bacteria. Okay, so this is how much boundary-induced torque you can have. We know that at low Reynolds numbers, there's no inertia, so you have to have your force balance and torque balance. You cannot have any unbalanced torque, unbalanced force, because there's no inertia. So what can balance this boundary-induced torque? We argue that the flexible hook that I showed you early on, when you have this torque on the hook will be bended, and this bend hook will provide actual torque that balances the boundary-induced torque. We assume the hook is flexible and follow this linear spring behavior. Theta 0 is essentially the wobble angle without this particle. and theta is the new wobble angle when you bend the particle, bacterial body, because the particle. And there's a bending stiffness, K, as a constant in front of that. Now you can balance this through torque. At low Reynolds number, the torque must be balanced, right? You balance this through torque, that's allow you to get a relation between size ratio of the particles as function of wobble angle. Remember from our experiment, I also show you that wobble angles anti-linearly relate to the swimming speed. Now you can change theta into velocity just using the empirical result. We just feed the experimental data with this linear line. Now we can convert theta into V. Now we can get a relation between velocity and size of the particle, gamma. There's only one fitting parameter in this whole equation here, which is the K, that's a bending stiffness of the flagella hook. Now you can feed your data with the existing data, with the experimental data, that's a solid line I have here. When you feed that, you find this constant to be 7 times 10 to negative 19 Newton meter. And Howard Berg has done this measurement very early on using optic tweezers. They found this value should be on order of 5 times 10 to negative 19. So it really match each other well. Okay, now next I have to tell you why this also applies to polymer. We know that if you have a polymer in fluid, you generate shear flow. The polymer coil typically behave in the non-draining limit, like Zim model, if you know polymer physics, meaning that when the fluid passing through the coil, it will never penetrate into the coil. Actually, it will go pass around the coil. And this radius here is a hydrodynamic radius of the coil, give you no slip boundary condition, essentially. And that's why coil behave like a particle. The difference is that you use a real size of particle, but you use the radius, hydrodynamic radius of the coil at the size of the particle, as a particle size. Now that's why they can collapse data together. Okay, the last thing that we need to say is we use this empirical relation between wobble angle and velocity. Can you actually predict how wobble angle determine the swimming speed? Theoretically, this sounds pretty straightforward, right? Because when you wobble, you swim along this helical trajectory and your projection into the swimming direction will get smaller. So the idea is pretty straightforward, but due to the calculations, a little bit tedious. I will go quick on that. If you're interested, I will list the reference of our paper later on. You can go to detail. The general idea is that you have to consider rotation of the flagella along one axis and rotation of the body along the different axis with a misaligned angle we call alpha. And then you have to balance the torque and force on the body and also flagella using what they call Purcell's model. Very early on, this is a classical model for flagellate bacteria. When you have that result, essentially you can predict how bacteria swim and what's the helical trajectory it has. And then you can do the projection to calculate the swimming speed. Again, I go very quick, but the bottom line here is that you can predict how velocity changes with wobble angle, and solid lines are result without any fitting parameter. Essentially, you can explain what happens in the system. Okay, so let me summarize the result we have in the first part of our talk, and I will go to the second part. So I'm basically telling you that there's the swimming behavior of bacteria in dilute colloid suspension is quantitatively similar as bacteria swimming in dilute polymer solution. And there's a common origin of the speed enhancement in both systems. And our result essentially telling you that when you go to the size of bacteria, it's very important to consider the discrete nature of the complex fluid. You cannot treat the polymer solution as a continuum medium anymore. You really have to consider of each individual coil particle interact with the bacteria through Newtonian fluid, through hydrodynamic interactions. And the second part of our study, we show you the origin of bacterial enhancement and also give you a model to showing how bacterial wobbling changing the swimming speed. And someone read the News and Views article for us. Actually, this captured our result really well. So this one picture is going to be your take-home message. Essentially, what happens is that when bacteria away from the particle, the wobble badly, going through this helix with large radius. But when the bacteria swim next to a particle, this helical structure get narrow because of hydrodynamic interaction. When bacteria swim more straight, they get higher speed. That's why they swim faster. The result is published last year. So again, if you're interested in all the detailed calculation, you can go there to see the detail. Okay, so that's the first part. When I talk to here, usually in the in-person meeting, people will ask me, Okay, now you talk about how bacteria interacts with the particle. How about two bacteria? Are they going to interact with each other? And do you see speed enhancement or not? Actually, people think about that, right? This is someone else's work published in PL about 5-6 years ago. And you can see that when you have two flagellar bacteria, in this case, different type of bacteria, but they use a similar way to swim, you can see bacteria actually bundle together. There's certainly hydrodynamic interaction between bacteria that allows them to form a pair to swim together. Now, in my research, I'm interested in more than two-part bacteria. We are interested in what happens when you have very high density of bacteria. You put them together in fluid, how they're going to collectively swim. Now, next, I'm going to show you a video of a high density of bacterial suspension. It's a real-time video and just demonstrate this collective behavior. So this is the next one here. Again, I'm not sure online, especially if you're in Australia, how smooth of your video. So if you do see smooth videos, you can see this beautiful jetting and vortex structure. If you freeze one frame, you have internet problem. If you're just stuck on one picture, it's really like Van Gogh's painting, Starry Night, right? You have this kind of jetting structures. Of course, a lot of people here study turbulence. If you compare this structure with the classical turbulence, they are very similar. They have this chaotic kind of coherent flow structure built into the system. But apparently, in this case, Reynolds number is much smaller, right? Reynolds number is 10 to the -5, no inertia in the system. By the way, the video I show you there is real-time video, and each of the black dot and white dot, as you can also see from this picture here, it is one E. coli. So you can imagine this, you can see that the density of E. coli is really high. Okay, actually, people also call the structure as a bacterial turbulence, right? There's many studies on this. So one thing that we ask in our lab is how does bacterial turbulence emerge, right? I show you early on with fluorescent microscopy with the dye flagella, you can see individual swimming behavior of bacteria. We know bacteria just swim individually with this random motion. When you add enough bacteria into a system, they start to show this collective flow behavior. It's very similar actually to fish schooling and bird flocking that you see probably on YouTube videos, right? How this happens? There any phase transition or crossover in the system? To study that, we design a genetically engineered type of E. coli where their swimming behavior can be controlled by light. So typically E. coli live in our gut. They don't care whether it's day or night. So they don't care about light. But with this genetic engineering, so the swimming speed of bacteria can be controlled by light. As I show you here on this data plot, here the velocity, the light intensity is controlled by the voltage on the light source. So I use the voltage here. As you can see, when increasing the light intensity, the swimming speed go higher. And the response is really fast. When switching the light intensity from 3 volt to 9 volt, you can see the swimming speed of bacteria just essentially in one second is already changed. Now, I prepare my sample without light, or with very low light. So you can see the suspension, but bacteria does not swim. Now at T equals 0, I start to turn on the light. When I turn on the light, you can see bacteria start to swim. And when bacteria start to swim, you begin to see this turbulent structure. So essentially we can turn on the turbulence on demand, right? Whenever we want it, we can have this turbulent structure. As you can see, the turbulence now form. The red arrow is the PIV result. As you mentioned, a lot of you are assume doing PIV. This is essentially the PIV of the fluid field. Okay. So with that, we can build up this phase diagram. There's 3 control parameters here. The first one is a fraction of swimmer. So we mix a swimming bacteria with non-swimming bacteria 100% when F equal to 1, meaning that all the bacteria are swimming. When F equals 0, meaning that all the bacteria are non-swimming. And we're also changing swimming speed by control the light intensity. And finally, we're changing the density of the bacteria. You get this phase diagram. The red dot is a turbulence regime, where you see this collective coherent flow structure. And black dot here is a random swimming. You don't see this coherent structure. Essentially, you have this crossover from random swimming to turbulence. Again, if you're interested in the standard, you can ask me a question after my talk. Otherwise, you can see this paper published 2 years ago. Okay, now, can you predict the phase boundary, right? Can you understand where this transition happens? Now, to understand that, you have to know the flow field around single swimmer. For E. coli, to simplify this picture here, you have flagella pushing the body, as I mentioned earlier on, and in the far field, away from the bacteria, you know the force field should be the flow field should be a force dipole flow field. Essentially, you have a force of the body and force of the flagella. They have to be equal to each other, force-free condition, but in the opposite direction. And this is a force dipole flow given by this 1/r² dependence. So U goes as 1/r² as confirmed by experiment. So this is the flow field one bacteria generates. Now let's think about two bacteria next to each other. How one bacteria influences another bacteria, right? If I have one bacteria swim nearby, that will be my bacteria along this blue trajectory, and the dipole flow will rotate my neighbor, which is this bacteria, vertical bacteria. And how much is rotate is given by this formula here in the red color. This is a far field dipole flow rotation, okay? Kappa here is essentially the dipole strength, which is known from the force of the bacteria, has been documented in biology for a very long time. Now, I add the actual order influence. So if the bacteria is self-swimming, it generates dipole flow, but dipole flow stretching this hard sphere particle. And this stretching of hard sphere particle will reflect back, give you the reflection flow. The reflection flow is weaker, so go as 1/r6. Again, kappa is the strength of the dipole flow. And this happens over a certain time, tau, which can be considered as a tumble time on order of 1 second. Now, this is basically how much hydrodynamic rotated. And on your own, bacteria also have this random rotation, and there's the effective diffusion coefficient we call dr. Now, the argument is like this. When the rotation caused by hydrodynamics is equal to rotation caused by random rotation, this will give you the critical radius. Beyond that radius, random motion will dominate. You will get random swimming. Below that radius, hydrodynamic will win out. You're going to have a collective swimming. So once you get this critical radius, you get a critical density. When your density is higher than that, you get a collective swimming. When your density is smaller than that, you get random swimming. So using this a simple, essentially hand-wavy argument, you can back up the concentration, the critical concentration of the bacteria, which is NC here. Above this NC, again, you see a collective swimming. And this essentially agree with our experimental result really well. The dot, the simples, is our experiment. The black line is our theoretical, hand-wavy theoretical result, right? So people has done this early on using more systematic way. They use kinematic theory, mean field theory to calculate the critical density of bacteria. This example here is by Don Koch at Cornell. That's actually give you the dashed line here. Again, agree with our experiment real well. This is in 2D. In 3 dimension, actually I already show you the result. This is a color surface, essentially our model prediction. You can see you can determine the transition between turbulence and random in this case. Okay, so far this is what we understand. The last part I will say something that is a really open question. I don't know the answer, but I think this is an interesting question worth to discuss with you. So what is the underlying orientational order of bacteria in bacterial turbulence? As I said, the structure that you see there is very similar to fish flocking, fish schooling and bird flocking, right? Actually, people consider this together as active fluid. There's many very interesting results recently on active fluid and review article. So people essentially put active fluid into different categories. There's a dry active fluid like fish and bird, or there's a wide active fluid like bacteria. There's also nematic active fluid where the bacteria can lie or the particle can align in nematic fashion, or polar active fluid where particle all align polarly. So people generally think that bacteria in bacterial turbulence just align like bird or fish. They align polarly. So they always put swimming bacteria as a polar, wide, active fluid. But we know that even though bacteria has a head and tail, clearly it's polar, right? The head is different from tail. It can align polarly, but it can also align nematically. There's actually no clear experimental evidence so far suggesting whether bacteria should align polarly or nematically. Everything that people have is essentially impression from bird or fish, which is big enough for you to see directly. People think that bacteria behave like fish and bird, but really there's no evidence for that. The question is that does really bacteria behave like a polar fluid, or they may just align nematically? Let me show you some experimental evidence on this. First of all, it's a structure of turbulence, structure of a turbological defect. Second is how bacterial turbulence show up, as I showed you early on. Third is macroscopic flow behavior. Last one is density fluctuation in the system. Topological defect. One clear signature of nematic alignment is that you can have this half integer defect, positive 1/2 or negative 1/2 defect. If you see positive or negative 1/2 defect, clearly your liquid is nematic liquid, because for polar liquid, you cannot go only halfway and go back to the same position. But the problem is that with so small particle, very small aspect ratio, with very high swimming speed, as you show here, and also three-dimensional flow, it's very hard for us to see any defect structure in the system. Actually, this is like turbulence. There's really no defect structure you can see in this flow. How about transition to turbulence? As I showed you early on, this transition can be predicted by dipole flow, far field dipole flow. And if you think about dipole flow is perfectly symmetric with respect to head and tail. There's no difference between head and tail. Essentially, the nematic symmetry is sufficient to explain the transition. But from that, it seems that the bacteria should be a nematic fluid. Furthermore, how about macroscopic flow structures? We measured a couple of things. First, we measured the correlation length, velocity correlation length. Essentially, you have PIV. You can measure how the two velocity correlated as function of size, as function of the length, distance. And we do that for system of different size. We essentially do find our bacterial turbulence in this Healy-Shaw geometry, and we measure how the correlation length changing with the gap thickness between the two plates. We found that this correlation length is increasing linearly with the gap thickness. Okay. And this is very similar, by the way, to the bird flock. If you have bird flock, they actually also see that the correlation length changing with the flock size. And there's some suggestion that a biological system poses as a critical point, criticality, but that's a different topic, probably very controversial. I'm not going to go detail on that. If you're interested, certainly you can read this paper here. Second feature is you can have velocity correlation. You can do Fourier transform on that. For people doing turbulence, especially a lot of people here, this is probably a standard thing for you, right? When you have a point-point velocity correlation, you do Fourier transform, you get energy spectrum. You get a scaling law, right? For turbulence, you get a Kolmogorov scaling, famous scaling. But for active fluid, you also get some parallel behavior at low K and at intermediate K. So intermediate K gave me -3 power, low K gave me a flight, essentially. And this is actually very similar to the prediction of active nematic fluid. But active nematic fluid, the most unstable mode is when K goes to 0. That means that the length scale should determine by the largest possible length scale in your system, which is system size. That's telling you the correlation length should go as the size of the system. Also, someone do the calculation show there's universal scaling for energy spectrum of active nematic fluid. Actually, the power is very similar to what we see in 3D system, -3 and flight, -3 and flight. In comparison, if you go to the most well-cited theory of active polar fluid, it actually predicts an intrinsic length scale of 50 micron, which is independent of system size, and the energy spectrum also show a peak, which is different from what we see in our experimental system. Lastly, you have density fluctuation in the system. So this is essentially optical microscopy. You can look at the turbulence flow, but you also see that there's a dark region and bright region. Dark region is essentially high concentration of bacteria, bright region is essentially low concentration of bacteria. And there's a strong density fluctuation, which depends on the system size. And this density fluctuation can only show up when you have a polar type of fluid. So the density fluctuation demonstrates that there's probably also some polar component in the system. So the conclusion is that, by the question I posed here, whether bacteria behave like fish or bird as an active polar fluid or as nematic fluid, one famous example is the microtubule suspension. The question so far is controversial. It's not certain. So if you look at the defect structure, we cannot tell too much. Based on the route to bacterial turbulence and macroscopic flow structure, bacteria should behave like nematic fluid, but based on density fluctuation, it should also show some polar structure. So with that, we run some very preliminary data so far. So basically what we do is that we dye a very small fraction of bacteria in our system and trying to measure their swimming speed and see how they align. I mean, just skip all this detail here, just tell you the final result. What we found is that when we increase in concentration of bacteria, when the bacteria go further into the deep turbulence regime, we plotting the probability distribution of alignment of two bacteria. As you can see, the alignment go more and more stronger along 0 direction, 0 degree. It seems that bacteria actually is a pretty strong polar fluid when you go higher concentration deep into the turbulence regime. With that, I really want to summarize because I think I'm right on time, 15 minutes. So I show you two parts. The first part, I show you how single bacteria swim in complex fluid. The second part, I show you the collective behavior of bacteria swimming. And I show you there's a hydrodynamic origin of the collective flow. And then I pose the open question here. What is the underlying orientation symmetry of bacterial turbulence? Hopefully our new result or continual research will tell you the answer in the near future. With that, thanks for your attention. I'm happy to answer any question that you have.
Thanks, Xiang. That was an amazing talk, super interesting. I have some burning questions, but I hope others would have as well. So I'll open it up for questions. Anyone from the audience, please feel free to unmute and ask your question, or alternatively, just type it in the chat and we'll read it out for you. Yeah, Yon, go ahead.
Yeah, I have a question about the first part of the talk. Thank you. It was a lovely talk.
Thank you.
You made the comment a number of times that there was no inertia. So I do work, I suppose, on particle migration, inertial particle migration. So we know that for finite size, small particles in viscous flows, they do move because of inertial effects.
Mm-hmm.
So why can you exclude that here? Just wondering, I guess maybe particle size or something.
Yeah, so I don't know exactly the Reynolds number that you have there. For single bacteria we have, because we know the size of the bacteria, we know how fast it's move, the Reynolds number 10 to -5.
Yeah.
Yes, you may still have very small inertia, but typically in the field, when people study bacterial swimming, they use the Stokes law, right? Stokes equation. There's no inertia in the system.
Right. So are the particles always smaller than your bacteria, about a micron, you said in?
Yes, the largest particle we use is 1 micron, so which is on same size of the bacteria.
So when you make your colloids, is it only 1 micron or smaller colloidal particles or bigger?
Yeah, exactly. We go from 100 nanometer all the way to 1 micron. The issue with the bigger particle, yeah, inertia could be a problem, but we haven't really think about that. Very good question. So the issue of bigger particles, the sediment really fast, right? 1 micron particle give you enough Brownian motion that at least over the length of the experiment, we don't see too much of sedimentation. When you go to 2 micron, 3 micron, over the time experiment, like 30 minutes, you can clearly see the density gradient between the bottom and top. And we cannot maintain the constant density of the colloid in our system. One thing we may thinking to do is apply some grant to NASA. They have some microgravity experiment. Maybe we can run experiment in the microgravity environment. Then we can go even higher particle size to see how bacteria interact with those bigger particles. And in that case, probably the inertia will play a role.
Yeah, okay. My guess is that, I mean, I've worked with neutrally buoyant particles, so there's no gravity effect. But
yes,
when you get 1 to 5 microns, then you do start to get some inertial effect. In that they'll migrate. The particles will migrate from streamlines. So yeah, it's interesting. But it might just be your particles are too small. Thank you.
Yeah. So we did think about the density match of particle, but mainly is because we are a little bit lazy on that, right? So when you density match the particle, essentially, I mean, a lot of colloid technically, you just changing the solvent, right? Mixing some even glycerol or some other type of fluid that the DMSO, for example, that's probably low density. So you go to different solvent, you can density match particle. But the issue with that is that a little bit challenging for this is that you have to have a solvent that is biocompatible, right? E. coli has to be happy and swimming and not died in those solvent. I'm sure there's some combination of solvent you can find, and bacteria is happy at the same time you can density match the particle. But we are a little bit lazy on that, at least at this point. So.
Okay.
Yeah.
Thanks. Xiang, I had one. So We saw that when the volume fraction was about 4%, you had that high jump in the velocities, and after that, the velocities kind of decreased with increasing volume fraction.
Yes.
Right. But in your second part, you had kind of when you looked at velocity versus volume fraction, like I think phi c, you found a monotonic, a kind of decrease with increasing volume fraction. So where does this the critical where does that volume fraction of 4% come into the picture in the other graph?
Yeah, it's well. I'm not sure if I understand your question, but I think I understand. If I understand wrong, let me know. So I think if you're asking why you have 4% that you have the largest speed enhancement, general picture is this: that imagine when you have only one particle in your system, you encounter this particle, you will bend the flagella hook, so the body and the filament line better, they will swim straighter, they will get higher speed. But after you move away from this particle, you will have this bend finally relax back to the equilibrium angle. And there's a time for that, right? Because everything kind of bend, go back to the equilibrium condition in the viscous fluid, in water in this case. And there's a time for that. So even before you can go back to your original position, you encounter the next particle, you bend them again. So if you encounter one particle after another, you can maintain this kind of the banded state that allow you to have a maximal speed enhancement. Right, so for volume fraction percent, we did some very quick, order magnitude estimate, actually gave you the distance between two particles when bacteria encounter one particle, before it can relax back, we encounter the next particle. So you keep encountering this particle, you keep swim straighter, you get a maximal speed enhancement.
Oh, all right, okay.
Does that make sense to you?
Yes, it does. It does. Okay.
Because if you don't have, you only have one particle. Once you swim away, then the bacteria will just go back to the original wobble behavior, right? So you have to keep encounter or interact with particle, and there's an optimal distance between two particles. When you encounter one, before you relax back, you encounter the second. Before you relax back, you encounter third, and then you keep doing this, give you the highest.
Okay. Right. Okay, makes sense. So that 4% is where it meets that critical point. Okay. All right. All right. There's a comment in the chat. So Abanob says there might be similarity between the motion of these bacteria and honeybees.
Yeah, I would definitely copy this and and watch that later after this talk. So I will copy that, because I know if I leave the Zoom or you close the Zoom, this will be gone, right? So I will just put the notepad here and copy this. Thanks for the comments. I'm looking forward to see this interesting analogy between bacteria and the honeybees. As I mentioned, that this is a whole field of active matter is really hot right now. Go to soft matter community. A lot of people talk about active matter. The general feeling is that there may be some universal kind of laws that they control all those collective behaviors, not only animal, but also artificial swimmers. And there's some common features that you can study together with those systems.
Oh, yeah. Just another question from me. So I think in the first part, so what you say is that the hook stiffness is the most defining parameter, right? That's what connects the wobble angle with the velocity. Is that right?
Yes.
Okay.
And that's something that, when they read this News and Views article at the end, they didn't talk to us directly, but at the end, they suggest something that could be done in the future, because there's some genetic engineered bacteria where the flagella hook, you can control how flexible of the hook. You can have a stiffer hook or more flexible hook. The question is that when you have those type of bacteria, how this swimming enhancement behavior change. That would be one nice kind of study on this.
Oh, okay. Do you think that
you can biologically control how stiff of the hook? Then you can see how this hook stiffness or the bending stiffness changing the behavior.
Do you think that has some relation to the human swimming in water and the viscous solution?
Probably not, right? There's some comments there. humans swim at the Reynolds number 10 to the 6, right? It's really, I mean, the way that we swim is really because of inertia, right? So if we swim in water, we just push, we always go forward.
Oh, yeah.
Yeah, for bacteria, you know, if you're rotating, you know, of course you go forward. But for algae, for example, you do breaststroke, algae do breaststroke. It has to go backward and forward and backward, forward, because there's no inertia. Right? I mean, if there's no inertia, in our case, when we pushing, let's say we do breaststroke, we pushing, when we retract our arm, we have to go backward.
Right.
The only reason that we don't go backward is because when we push, we already have a strong inertia that's push us forward. Even when we retract our arm, the inertia is still strong. We're still going forward, right?
Yeah.
Right. So inertia is super important for human swimming.
All right. Yeah. Makes sense. Okay. So I think if we don't have any more questions, please join me in thanking Dr. Xiang for an excellent talk. And I hope we all have something to take away from this.
Thank you. Thank you for the question and suggestion.